Hello, good morning everyone. This is Rohini Prasad here. Today, dear students, we'll study about classification. Now, when we talk about classification, the definition which you can see on the screen is it is a system of arranging living organisms into groups based on their similarities and differences. Now, when we talk about classification, a good example you can take of your library. You just think that if books are piled up without arranging them into the shelf, and now if it is asked that you have to classify each and every book and you have to keep it in the shelf, so how you all are going to do that? First, the first you all are or in fact, if I am told about it to do, first I am going to sort down the book according to the subjects. Suppose bio, physics, maths. I'll pile up the books according to the subjects, and then again I'll pile it up according to the classes, like class 1, class 2, class 3, 4, 5, 6, 7, 8, and then according to the writers or publishers, I'm going to pile up the books, and then I'm going to arrange it into the shelf. Similarly, when we talk about classification, it is just similar to that only. Whatever the similarities are there in an organism, that we have to see, and what are the differences in an organism. Now, for classification, if we take an example, you can classify plant and animals. Plant is different from animals, and animals, the features of animals are different from the plants. Like plants cannot locomote, but animals can locomote from one place to another. So that is a key difference between the two things, between plant and animals. Therefore, by the definition, it is clear it is a system of arranging living organisms into groups based on their similarities and differences. This is known as classification. Previously, scientists were working on this classification, and they found out and they classified the whole system into plant and animal only. But further, as discoveries were made, certain things were kept in mind and then they were classified. Now, what are the advantages of classification? Advantages of classification, when we talk about why we need to classify any organism. Characteristics of all members of group can be understood. Means each and every organism on the earth is having its own characteristic. Like amoeba is a unicellular organism, human being is a multicellular organism. So this is a key characteristic of these two organisms. It makes the study systemic. This means one by one each organism's characteristics can be studied, and it can be systematically classified or categorized into different groups. It helps in identifying different organisms and placing them into a particular group. Now, this classification help us to identify organism, which means like we can identify birds, insects, human beings, animals. Similarly, these organisms can be identified by their typical features. Like birds can fly, okay. Insects, in certain insects can fly, certain insects cannot fly. So in insect also classification is there. They are also categorized into different groups. This is about advantage of classification. Next, when we come classification of living world, as I discussed before, that many scientists classified the living world into kingdom Plantae and kingdom Animalia. But further, when studies were made, they came to know that other than this, these can be classified into other groups. So first two system classification came, then three kingdom classification came into existence, then four kingdom classification came into existence. But the scientist named Robert Whitaker, he proposed 5 kingdom classification. According to him, the whole living world is classified into kingdom Monera. The most common example of kingdom Monera is bacteria, kingdom Protista. The common example of kingdom Protista is amoeba, kingdom fungi. Common example is mushroom, kingdom Plantae. All different type of plants on this earth come under kingdom Plantae. Kingdom Animalia, different varieties of animals come under kingdom Animalia. First, we are going to study kingdom Monera in detail. What are the characteristics of kingdom Monera? What are the examples? What is the use of kingdom Monera? What are the disuse of kingdom Monera? So first, we will study about kingdom Monera. Now, when we come to kingdom Monera, kingdom Monera is having a few characteristics. You can say that under kingdom Monera, the most common example is bacteria, and it is a prokaryotic organism. Prokaryotic means the organisms which are primitive in nature. Primitive in nature means they are the oldest or primitive form of organism on the earth, the first organism, or you can say before all the animals, before all the plants, before Protista, before fungi, kingdom Monera came into existence. They are unicellular. They don't have any membrane bounded cell organelle, like they don't have mitochondria, Golgi bodies, endoplasmic reticulum. These cell organelles are absent in kingdom Monera. Then single circular chromosome is present there, means they have only a single stranded DNA is present there, and they reproduce by binary fission. Binary fission means they split into two. The cell will divide into two, so that is known as binary fission. Now next, when we come, there are different forms of bacteria. On the screen you can see circular, rod shape, curved form, and other shape of bacteria are there. Circular shape bacteria are also called as coccus. Then rod shaped bacteria are called as bacilli. Curved shaped bacteria are classified into spirilla and vibrio. Now when we talk about circular shaped bacteria, it can be in two pairs, in chains, or in clusters. When they are in pairs, it is called as diplococcus. Then in chain, it is called as streptococcus. In cluster, it is called as staphylococcus. Similarly, rod-shaped bacteria is called as coccobacilli, streptobacilli, and mycobacteria. Curved form of bacteria, when we talk about vibrio form of bacteria, it is a comma-shaped bacteria. Spirilla is a spiral-shaped bacteria, and Spirochaetae is also a spiral form of bacteria. Other shapes are helical, which is called as Helicobacter, club type of bacteria, which is Corneobacter, and Streptomyces, a single thin long elongated structure of bacteria. Next, we come to structure of a bacterium. A typical bacterium cell, how it looks like, that we come to. When you see the structure of bacterium cell, this bacterium cell is devoid of nucleus. They don't have nucleus, so the DNA material which you can see, it is scattered, or it is lie like that only. It is not enclosed in any kind of membrane bounded cell organelle. The outermost cell wall of bacteria is made up of protein like material, and a plasma membrane is also present there. Then a proteinaceous gelatinous cell wall is present there, which is called as capsule, which protects the bacteria from other organisms. Nucleolus, mitochondria, and plastids are absent. Then reserve food is present, which is called as glycogen. And when we talk about bacteria is very small in size, micromunes in size, mu meters in size. Okay, and now when we talk about useful forms of bacteria, there are certain useful forms of bacteria which is used in our day-to-day life. Like without good bacteria, bad bacteria would completely take over the body, so we can have various kind of diseases. Therefore good bacteria are there, and some good ways to use bacteria includes sewage treatment. Sewage treatment means when the sewage from the household activities or from hospitals or from other municipal sectors is sent into the sewage, there these bacteria work on those sewage plant and decompose them, and then further it is discarded. Then recycling of dead organisms, the dead organisms which are present there, these bacteria petrify those dead organism and decompose the dead body of those organisms. Then producing and processing food, various food and are being processed. Such examples, when we talk about processing of food, you can say sour cream, yogurt, buttermilk, cottage cheese, pickles. These are the process, these are the foods which are being processed by the bacteria. One example, you can say lactobacillus bacteria is used for curdling of milk. That means formation of curd from milk. How does this happen? It converts the milk sugar into lactic acid, giving the sour taste to the curd. Then certain bacteria like Acetobacter ferment fruit juices into vinegar, and commonly we call it as acetic acid. Then these bacteria are also used in producing chemicals and fuels, mining minerals and petroleum, cleaning up oil and chemical spills. Other than this, when we talk about bacteria, they help in tanning of leather, then retting of fibers. Now when we are talking about sewage treatment or recycling of dead organism, when you talk about this can be used for preparing manure also. Okay, then other thing, biogas is also there are sewage treatment. When we talk about, there are sewage plant in big cities where the collected human excreta is decomposed by sewage bacteria. The inflammable gas produced in this process is used for cooking purpose, and the liquid and solid product are used as manure. So this is how sewage treatment bacteria are very useful in sewage treatment. Then certain bacteria are there, like rhizobium is there, which are present in the root nodules of the plant, which convert atmospheric nitrogen, nitrates into atmospheric nitrogen for the plant. Here is a small video which will just make it clear to you. Just see the video.
Also require a lot of nitrogen to make proteins. So they absorb nitrogen continuously from the soil, due to which the amount of nitrogen becomes deficient. As you all know, that there is 78% of nitrogen present in our atmosphere, but plants cannot use it in the manner directly as they can use carbon dioxide. They require nitrogen in soluble form. The bacterium called rhizobium can take atmospheric nitrogen and convert it into a soluble form. These bacteria live in the roots of gram, peas, mung beans and other legumes and provide them with nitrogen. In return, the plants provide food and shelter to the bacteria.
Next, when we talk about, bacteria is very important in medicine producing also. It produces various kind of medicines. Just see the video which I am showing to you.
Useful role of bacteria in medicine. Welcome to the new session of Economic Importance of Bacteria and Fungi. In our present session, let us learn about useful role of bacteria in medicine. In medicine, bacteria are used in different ways. But the role of bacteria is vital in the production of antibiotics, serum, and vaccines. So, now let us learn about the role of bacteria in production of antibiotics. Generally, most of us, on visit to our physician when we are sick, might have heard about antibiotics which are prescribed as medicines for our sickness. Did you ever think what antibiotics are? In definition, an antibiotic is a chemical substance produced by a living organism which can stop the growth of some disease-producing bacteria and fungi. Can you list out some of the antibiotics you heard so far? Most of us had heard about penicillin. Penicillin is an antibiotic produced from the bacterial sources. In similar way, streptomycin, chloromycetin, and tetracycline, etc. are all bacterial-sourced antibiotics. In medical history, penicillin is the first produced antibiotic. To the fact, penicillin is not the product of bacterium, but it is the product of mold, fungus. In the year 1929, the scientist Alexander Fleming was growing some bacteria in a culture, but he observed the culture was destroyed due to accidental contamination of a mold, Penicillium notatum. Thus, he discovered that some substance that is produced from the mold, fungus, had suppressed the growth of many bacteria in the culture and named the substance as penicillin. This penicillin is used as first antibiotic for medical use. In similar way, the scientist Salman Waksman discovered another antibiotic named streptomycin. This antibiotic is a bacterial source from Streptomyces gracius. Now, let us learn some of the known antibiotics and their sources and diseases treated by them. Antibiotic. Source organism. Disease treated. Penicillin. Penicillium chrysogenum. A mold. Disease treated. Tetanus, diphtheria. Streptomycin. Streptomyces gracius. Disease treated. Tuberculosis. Chlorotetracycline. Streptomyces aerofasciens. Disease treated. Typhoid. Erythromycin. Streptomyces erythraeus. Rickettsial fevers. Now, let us learn about criteria of a good antibiotic. A good antibiotic should be able to kill a variety of disease-producing microorganisms. A good antibiotic should not produce any undesirable side effects on usage of it. A good antibiotic should not kill normal bacteria in the host. Some other uses of antibiotics. Antibiotics are also used as food preservatives for fresh meat and fish. Antibiotics are used for treating animal feed. Antibiotics are also used in controlling plant pathogens.
Now, as we have studied about the useful effects of bacteria, there are certain harmful effects of bacteria also. It gives you disease, it can rot your food, they lose the life of plant and animals. That means it can cause disease to plants also and animals also. What now? Disease caused by bacteria. There are certain diseases which are caused by bacteria, like streptococcus. Mode of transmission: direct contact with respiratory and other body fluid. Disease caused: sore throat, fasciitis, toxic shock syndrome. Damage: cell, tissue, and muscle destruction. Symptoms: sore throat, rash, and cellulitis. Then salmonella, it the mode of transmission is food. Disease caused is salmonellosis. Then damage is endotoxins. Symptoms are diarrhea, fever, and abdominal cramp. In simple words, we can say that it is food poisoning. Then next is Vibrio cholera. Mode of transmission: water. Disease caused cholera. Damage: enterotoxin, watery diarrhea, abdominal and leg cramp, and fever. Then treponema, this mode of transmission: sexual contact. Syphilis, disease caused. Damage: destruction of epithelial and endothelial cells. Then symptoms: small paleness ulcer, rosy skin, paralysis, blindness, and dementia. The next is Yersinia pestis. It is the mode of transmission is insect, and disease cause: bubonic plague. Damage: bacterial growth in lymph node. Symptoms are fever, headache, chills, weakness, and swollen tender lymph glands. These are the diseases caused by bacteria. I hope you all have understood about the basic concept of classification and about kingdom Monera, the structure of bacteria, the useful and harmful effects of bacteria. Thank you. Stay home and be safe.